And the first semifinalist in the overgraduate section is Bielefeld. Welcome. Take it away.
Hello everyone, this is Lucas, I'm Tommy, we're part of Bielefeld's 2013 item team, and on behalf of the team I'd like to start by saying thank you for having us here. I did not expect to stand here today, otherwise I might have skipped a few drinks. But now to our project. It's called Eco-Electricity because we're using E. coli to produce electricity in an ecologically friendly way. And this is because, as everyone probably knows, supplying all people in the world with sustainable energy is one of the great challenges of our time, and this is because many ways of energy generation have negative side effects like CO2 output or generation of radioactive waste. And also, energy is rarely produced at the time and place you need it, so it has to be stored, for example, in batteries. And batteries are made out of scarce materials like lithium. They are not easy to dispose of once they're broken, and also they take quite some time to charge. And one way to tackle all these problems is using fuel cells. A fuel cell works quite similar to a battery. It's split up into two chambers, and to the left in the anode chamber, a high energy compound is oxidized and electrons and protons are freed up in the process. And in the cathode chamber to the right, another chemical is reduced with these electrons and protons. And the chambers are separated by a proton exchange membrane, and protons can freely travel between the chambers, but the electrons can't. So they have to travel to the anode and through this wire connecting it with the cathode. And this way an electric current is generated. And what we did is we placed E. coli in the anode chamber to act as a biocatalyst. And E. coli metabolizes organic substrates and also frees up electrons and protons in the process. So it also can, in theory, donate its electrons to this anode. And fuel cells have some general advantages. They supply the energy right when you need it, and they're easily scalable, so they can fit in your pocket or even inside a cell phone. And microbial fuel cells share these advantages, but they have one major advantage over the normal fuel cells. They can use a variety of different substrates like different kinds of sugar or glycerol and even waste, because it still contains many high energy compounds. So research teams around the world are already working on the subject of microbial fuel cells, but usually mixed cultures are used for these purposes. They're isolated from the deep sea or from wastewater, and this is because they are very good at transferring their electrodes to an anode, and E. coli unfortunately isn't. But we still chose to work with E. coli because it grows very fast and it's also quite sturdy regarding cultivation conditions. And in these mixed cultures, it is usually unknown which strains really are inside these cultures. So they become quite unpredictable, and this also raises safety concern if you do not know if pathogenic strains are part of your culture. And the main advantage of E. coli, of course, is its accessibility to directed genetic manipulation. So with the powers of synthetic biology, we can transfer the abilities of these mixed cultures over to E. coli, so they become better at giving their electrodes to an anode. So our project can be split up into two levels. The hardware level, where we started by constructing a microbial fuel cell, because of course we needed this to conduct our experiments in, and also we wanted to make this MFC technology available to the IDEM community, just like we did with our BioBricks. And we also had to establish some standardized measurement parameters so we could compare all the data we generated. And second is the genome level. As I said, E. coli isn't very good at transferring its electrons to an anode, and this is because the electrons are not able to cross E. coli's membrane, and also once they have crossed the membrane, there is nowhere for them to travel to the anode. So we had to enable both of these functions, and also we wanted to implement a biosafety system, so this would allow for a real-world application of our fuel cells. And I'll start by explaining what we did on the hardware level. we started by constructing a very simple fuel cell out of just two film canisters and part of a centrifugation tube, and this worked as a fuel cell, but it is not anaerobic. And this is very important because if there's oxygen available, coli will always just transfer its electrons to oxygen and not to an anode. This is why we built this second model. This is inspired by a design published by H.P. Benedetto in 1990. It consists of 4 plastic parts, you can see here, and the ones in the middle, we call them the frames. They make up the anode and the cathode chamber, and electrodes are placed in the middle of each, and they're separated by a proton exchange membrane. And this model worked pretty well, but it still had some limitations. We found out that we never knew how long it took for E. coli to use up all the oxygen in the anode chamber, and this made measurement very difficult. So we wanted a way to gas out the oxygen with nitrogen, and also it took a lot of time to refill the chambers because we always had to fully disassemble and reassemble the cell. And this is why we built this third version. It's basically the same design. The frames have been split up into 2 parts to allow for easier mounting of the electrodes, and it features tube connectors at the bottom to allow for nitrogen aeration, and also it allows us to exchange the liquids inside the chambers. And we also built a customized version of the cell for the Agent Team York because they were also working with microbial fuel cells. We still weren't content with this though. We used this for all our measurements, but as I said, we wanted to make this available to the whole community. So this is why we went to a local hackerspace in Bielefeld, and they showed us how to use a 3D printer, and they let us use theirs. So we spent quite some time developing a model which you can print out using a 3D printer, and the files are available at our wiki, and you can print them out by yourselves, or if you don't have access to a 3D printer, just upload them to an online 3D printing service. And all the materials needed for such a fuel cell can be obtained for less than €4, and it's even printed from biodegradable plastic. So now we get to the genome level of a project. As I said, this was about enabling electron transfer across the membrane and enabling the electron transport to the anode. And we had 5 different approaches for these problems: 2 for the transport across the membrane, 2 for the transport to the electrode, and nanowires, which could have potentially solved both problems. And we worked on all these 5 approaches, and we also met an expert on the topic of microbial fuel cells at a conference, and we consulted with him several times. And over the course of our project, we decided to focus on these 3 approaches because they seem to be the most feasible. And riboflavin and GLDA are about mediators, and I will now explain what a mediator is. It is a redox molecule that means it's able to transport electrons from a bacteria to the anode and travel back and collect new electrons. And we had some demands. Of course, it had to be soluble, and also it shouldn't be toxic, so our cells wouldn't die. And chemical mediators are available, like methylene blue, which we used for quite a lot of experiments, but they're not a real solution because they're quite expensive, and this is a problem especially if you consider the amount of energy a single fuel cell can generate. And now I will hand over to Lucas, who will explain what exactly we did on the genome level.
Thank you, Tommy. Now we have finished the engineering aspect of our project, and we are going on with the genetic optimization of E. coli in order to optimize it in order to an efficient electron transfer and electricity generation. As we have already here, we need mediators in a high concentration for an efficient transport of the electrons to the anode. But this admission of these mediators into our fuel cell from outside is very expensive. So we would like to produce these mediators by E. coli itself. And one possible mediator is riboflavin. We produce this by a heterologous expression of the riboflavin synthesis gene cluster from Chivanella oneidensis in order to produce this riboflavin. And after successful cloning and the first cultivation, we got an impression of the successful expression of riboflavin. In contrast to E. coli wild type, we could show a distinct yellow coloration for our riboflavin producing strain and a further enhanced riboflavin production when using a stronger promoter. To confirm these first impressions, we did just several experiments, and I would like exemplary to show you our fluorescence assay. As we can see here, we have no riboflavin production for our E. coli wild type after 12 and still after 72 hours. But in contrast to that, our riboflavin producing strain has a riboflavin concentration after 12 hours of 300 microgram per liter and a further enhanced riboflavin production after 72 hours with 4 milligram per liter. We confirm these fluorescence assay also with absorbance assay, high performance liquid chromatography and LC-easy MS. Besides the production of riboflavin as a possible mediator, we also used NADH as a mediator. Therefore, we overexpressed the glycerol dehydrogenase from E. coli. And this works, as you can see here. The glycerol dehydrogenase catalyzes the reaction from NAD+ and converts it. And converts it to NADH. And the most important fact is that we can transport with NADH as a mediator 2 electrons to the anode. To confirm this, we used a fluorescence-based NADH assay. And as we can see here, we have the lowest NADH concentration when using E. coli wild type. And further enhanced NADH production when using our GLDA producing strain. We can see an increased NADH concentration up to a maximal NADH concentration with a 160% enhanced in comparison to E. coli wild type. And this is for an increasing promoter strength. We used this in order to confirm it for electricity generation. And what we can see is a 40% average electric power generation for our GLDA optimized strain. After successfully production of these mediators, we had to come to a main challenge of our project, and that is the optimization of the membrane of E. coli. And of course, E. coli is able naturally to transport electrons with mediators produced by us across the membrane. But we have a very slow transport kinetic across the membrane, and that's the fact we are starting. We optimized this transport kinetic across the membrane by permeabilizing it with introducing porins in the outer membrane in order to enhance the transport kinetic across the cell membrane and to enhance electricity generation. We did this with using OPRF from Pseudomonas fluorescens, which is one of the largest porins on bacterial outer membranes with a permeability for very large molecules. And to get a first impression of the expression of these OPRF, we performed atomic force microscopy with the help of the physics department at University of Bielefeld. And what we can see here is a very smooth surface for E. coli wild type and an enhanced roughness when visualizing our OPRF producing strain, which only indicates a successful production of these OPRF on the outer membrane. In order to confirm this, we performed several experiments in order to a morphological and physicochemical characterization of our E. coli, of our optimized E. coli. Exemplary for this, I would like to show the MPN uptake assay with a standard measurement for membrane permeability. And we can see the lowest fluorescence for E. coli wild type. The fluorescence stands directly for the membrane permeability. And what we can see is an increasing permeability for our OPRF producing strain with an increasing promoter strength up to a maximal membrane permeability for our OPRF producing strain under control of T7 promoter, which is circa 100% enhanced in comparison to E. coli wild type. And using this in our microbial fuel cell, we could observe a 100% enhanced voltage. This orange curve shows our OPRF producing strain, and this is the E. coli wild type. And we have a very good optimization of electricity generation. Transferring this data into an average electric power diagram, we could see a 400% enhanced average electric power for our OPRF producing strain. With using this optimized strain in combination with our microbial fuel cell, we enable an efficient electricity generation. With this, we were able, for example, to light lamps. And the great thing is that further optimization is still possible. We have the possibility for a combination of these purines, for example, with our mediator production system. Now we have a functional approach with our microbial fuel cell by combination of the genetic part of our project with the engineering aspects. But now we want to go a step further. We would enable a real-world application. So we thought about a biosafety system. Our biosafety system is inspired by the iGEM team Paris Betancourt in 2012. We took one part of them, which was not functional, and improved it. And around this biobrick, we constructed a complete new biosafety system. The fundament of our biosafety system is the biosafety strain. We constructed it, and it is the alanine oxytrophic E. coli with a knocked out genomic alanine racemase. And so this strain is characterized by strictly dependence on the alanine, which doesn't occur in the environment. It's essential for membrane building. In order to complement with oxytrophy of our strain, we introduced the alanine racemase in our biosafety plasmid, because the alanine racemase is able to convert L to D-alanine. And additional, we have a second kill system, and that's our toxic gene product, the RNase bar, or called Barnase. We not only designed one biosafety system, we have 3 biosafety systems. We have the same working principle, but they differ in their regulatory system concerning their promoter and repressor. That's why they are called tether-alive, erective, and lack of growth. I would like to present you our best working one, erective. As a regulatory substance in our fuel cell, we use remnose. Remnose doesn't occur in that form in the environment, and so it's quite a good substance for our fuel cell. Remnose activates the remnose promoter, and therefore the repressor RSC can repress the PBAT promoter, and so an expression of the Barnase isn't possible, and the cell can survive. And beside this, we have a complementation of the oxytrophy by a positive expression of the alanine racemase, which converts L to D-alanine, and the cell can grow. In absence of remnose, we have no activation of our remnose promoter, and therefore also no repression of our Barnase, which cleaves the intracellular RNA, which leads to cell death. And beside this, we have our second kill system, the alanine racemase. This can't be expressed, and so we have no D-alanine production, which finally leads to cell lysation. So we have an active double kill switch system, which maybe enables a future real-world application. We tested our biosafety system with GFP instead of this Barnase, and we could indirectly prove the functionality of our biosafety system, and it works as expected. After constructing our biosafety system, I would like to sum up our project and show you our achievements. We were able to design, construct, and improve a microbial fuel cell, and we were able to gain electricity out of that. As you can see here in this video, we were able to use a small motor to make movement with a fan, and we were able to make electricity to light different LED lamps. Besides this, we printed a biodegradable and constructed it a biodegradable DIY MFC for making our approach accessible to the community. And of course, we built a lot of biobricks over the summer. For example, these porous biobricks for integrating in the outer membrane for a permeabilization of the membrane and an enhanced electricity generation. We furthermore produced riboflavin as a mediator substance and NADH by overproduction of the glycerol dehydrogenase. And we enable a future real-world application by introducing 3 functional biosafety systems. And furthermore, of course, we had several human practice projects. For example, we took part at different conventions where we presented our project. We organized with other German iGEM teams the Day of Synthetic Biology. We supervised and organized the Student Academy at Center of Biotechnology in Bielefeld. We had different contributions in TV and radio, and we helped other iGEM teams, for example, Team York, with constructing a miniaturized microbial fuel cell for them. So finally, I would like to say thank you to our supporters. Without them, a successful participation in this competition wouldn't be possible, and eco-electricity wouldn't be currently available. So finally, I would like to say thank you to you all. It's very great to be here today, and yeah, thank you very much for your attention.